Hello and good afternoon. My name is Shirin, and as been kindly said, I'm a specialist in the Center for Antibody Technologies, Technical University of Venoms. It's my first time in Oxford, and it's a pleasure to be giving you my talk on proteomics and histological assessment of an organotypic model of human skin upon exposure to nigricollis venom. So Naja nigricollis, or the black-necked speaking cobra, is one of the 7 species of African speaking cobras that are widely distributed in Sub-Sahara. And distribution in Naja nigricollis is seen in blue here, hopefully if you're not colorblind. African speaking cobras share an overall venom composition dominated by PLA2s and cytotoxins. And cytotoxins are a group of 3-finger toxin family that interact with and disrupt membrane of different types of cells in the skin and underlying tissue. They cause dermonecrosis and ulcers and non-healing wounds. And in severe cases, they may further develop into gangrene, necessitating limb amputation. So next photo is not easy to look at, but it showcases a case of dermonecrosis in a child. So in fact, out of the 400,000 cases of sequela that are attributed to snakebite envenomy annually around the world, it is estimated that roughly 6,000 to 15,000 amputations are happening only in Africa due to snakebite. So there are many different reasons for these high and alarming numbers. One of them is the way that we produce antibodies, which is through immunization of large mammals like horses. So as Professor Varro also mentioned, if a toxin is not immunogenic, then we wouldn't get a strong immune response in the host animal. And therefore, the general efficiency of antivenoms against locally acting venoms is low. And that's why the high numbers of local damages. So when I started my PhD, I decided to focus on the local damages, while my colleagues were passionately working with neurotoxic venoms. What I did, I used phage display technology to select for antibodies that could bind to cytotoxins from both Naja nigricollis and other African speaking cobras. After several rounds of selections, I had promising binders that could bind to cytotoxins from different species. But since I was the first one in the lab working with cytotoxic venoms, we didn't have in vitro assay setup in the lab. So in collaboration with another group, I set up a monolayer culture of keratinocytes, which are the outer layer of our skins, in the lab and a cell viability assay. So in this assay, after determining the IC50 of the venom, if I treated the cells with 2 IC50 of the venom, they were all dead. If pre-incubated it with an irrelevant antibody, it didn't do that much. But the discarded antibodies in the phage display could work, and cells remained 70 to 90% alive in this assay, which was very promising. And then I also, during my literature review, realized that there's very little information, as we already talked about, in different mechanisms and pathways that are affected by cytotoxic venoms and their effect in the dermonecrosis. Anyway, after getting these promising results in vitro, we went to in vivo experiments, and unfortunately, we didn't get in vivo neutralization there. which was very disappointing. But after that, I kept thinking to myself, if we want to comply with 3R principles of refinement, replacement, and reduction, then we're gonna need better assays, in vitro assays, for testing our antibodies and neutralizers before we go to in vivo assays. And that's why in my postdoc, I worked with a more complex in vitro assay. Here I made a 3-dimensional organotypic model where extracellular matrix was mixed with fibroblasts first, and then a layer of keratinocytes seeped on the top. And then we gave it time to differentiate into different layers. And finally, it was injected either with PBS for control or venom. So the idea was that check if this approach, if this model works for venom alone. And if it does, then we check it for the neutralization effect of antibodies. After confirming the establishment of the model, we also injected some mice to compare. And when we look at the sections under microscope, we see some inherent difference between mice and human, right? Because in mouse, we only have 1 or 2 layers of keratinocytes, while in human, we have between 8 to 10 layers. So there is a difference there in addition to the number of hair follicles in mouse compared to human. And when we look at the venom-treated part, we see both mild and severe effect in mouse, while in the organotypics, we see only severe damage. How do we define that? These stained parts on the organotypics show the healthy live nuclei of the cells, but here they are either gone or shrink. After this, we looked at the proteomics data of both. So here we also have some differences between different replicates. For example, this one, venom number 2, is more close to control number 1 compared to other venom treatments in mouse, or here in organotypics, we have a clear difference between the replicates treated with the venom. Anyhow, we found 2,500 proteins in mouse, 1,300 in organotypics, with more than 900 overlapping between the two. Next, we looked at the number of the proteins that showed increased abundance or decreased abundance upon exposure to the venom. And we had almost the same number of proteins in both data sets, but when we looked at the number of them that was shared between the two models, we didn't see that much of overlap. So one of the reasons can be that we have a protein that that has a specific function and works in a specific pathway, but the nomenclature is different between different species, and that's why we cannot see them here. Fortunately, we have gene ontology terms to look at, and they are proteins that are in the same pathways or having similar functions, but they may have different names. Here we see much more overlap between the number of pathways that have been affected between the two models. And here are just some of the GO terms. It's a busy slide, but here you see, for example, that apoptotic execution phase or response to wounding has been affected in both of the data sets. So if we look at the number of proteins that show increased abundance in both models, we see a complex interactome of all. But if we extract data based on pathways, then suddenly some pathways show up. For example, one of them is regulation of insulin-like growth factor that has been affected in both models, and phosphatidylinositol-3 kinase signaling pathway, and also keratinization, which is the pathway that leads to survival of the keratinocytes and wound healing. So there are so many links missing between these pathways here, but I can imagine that a hypothetical pathway inside the cell would look like this. So upon exposure to the cytotoxins with unknown mechanisms, IGF pathway is affected. So some protein inhibition is removed from the binding protein, and therefore IGF-1 is released to bind its receptor, and then a cascade of kinases are activated inside the cell, which together relay the message to the nucleus to increase expression of genes that are related to keratinization, and therefore leads to wound healing and survival of the cells instead of death. So if I want to conclude, we need better models to in vitro test our treatments before we go to mass studies. And unfortunately, due to lack of funding, we couldn't test this model for neutralization effect of antibodies and see how it's translated to in vivo results. But I can imagine that it's also doable there. And in the end, I would like to honor memory of Professor Keller, who was a main collaborator in this project. And we unfortunately lost him last August. And without his immense knowledge and expertise, this wouldn't have been possible. And also thank my colleagues in the Center for Antibody Technologies. With that, thank you.